Welcome back to our class. Today we're going to be talking about the June exam for 2025. Let's see what this district wrote. They're saying that let's go to the questions without wasting time. They are saying the cell wall of fungi are mainly composed of, they are mainly composed of what you call chitin, cellulose, protein, lignin. Still, all this is uh, protein-like component, protein and carbohydrate-like component. But the fungi is made up of chitin, which makes them to be much more protected. Then they're saying that question one, from this question to this question, we need to answer based on this. So we have what we call animal with the radiosymmetry. Radiosymmetry meaning that it can be divided in many planes, in many planes. So its answer becomes uh, B. So we're gonna have to see that uh, is B. And then if you look at structure, structure C, it shows that it cannot be divided into two, because when I divide it like this side A and side B are not equal. If I divide it like this, C and D are not equal, so it's asymmetrical. All this one, it is already showing it is bilateral symmetry. So the answer becomes B, as I said. So the D becomes the most correct answer. Then we go to the next question, the phyla to which uh, animal C belong. Animal C, animal C, which else? The sponges. Where do the sponges belong? I think this question is supposed to be the phylum means many phylum. So the phylum to which this belong, because it is one. So we are saying that those are sponges. Sponges. So the answer becomes polyphera. Yes. So 2 marks there. Then we go to the next question. They're saying that 1.4, 1.14 store from. So it means that that question is now done. Saying that a stored form of glucose, we call it in plants, we call it starch, and then stored form of glucose in animals, we call it glycogen. So since we are looking for the plants, so the answer becomes C. C becomes the most correct answer. So we have the structure of the chloroplast here. Where in this chloroplast would you expect to find? So even this question is showing you that from 1.15 to 1.18, we're supposed to answer based on that. So they're saying that where in the chloroplast would you find the light independent phase occurring? So we will find out that we need to find out that light, they're saying what? Where light independent, meaning that this is the dark phase. Dark phase. So we see that these are the grana, and then these are this is the cytoplasm. Remember, did I say cytoplasm? Stroma. This is the choroplast. Stroma. Remember that the dark phase occurs in the stroma. So I have to look for B, which is that, and then the answer becomes there. Which of the following statement about the light independent phase is correct? The light independent phase. It requires glucose. Only they're saying that only sunlight occurs. Light independent, no sunlight needed. Requires glucose. It only occurs when there's it does not need sunlight. Yes. The light independent phase, remember glucose is broken down into three carbon atom molecules. Three carbon atom, meaning that two of them, that is pyruvate. Eh? No, 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 no. That is no. Yeah. Remember that glucose is being produced. Glucose is being what? Is being produced in this light dependence. That's why I say that if they say that they require glucose, then it means that this is what is wrong. This is respiration, and that is glycolysis. All right. So the most correct answer here becomes D, and then we get 2 marks in there. They are saying that question number 7, which molecule is involved in light independent reaction? Light independent reaction. If you look at light independent reaction, oxygen, ADP, NAD, nope, oxygen, NAD, AD, and that is, we call this one adenine adenosine diphosphate, and then we have nicotinamide adenine dinucleotide phosphate with the hydrogen. So the which one is light independent phase? We need carbon dioxide. Yeah, we need carbon dioxide in the light independent phase. ATP and NADH, they are being produced. So D becomes the most correct answer because they said are involved, meaning that they're not saying they are produced or they're being used, they're saying they are involved. So D becomes the most correct answer. Where in the plant leaf cell does the light independent reaction occurs? Light independent reaction, the light independent reaction. So now you have to go back and you find out the light independent reaction. The light, did I say light independent? The light dependent, because we have already talked about the light independent. So the light dependent occurs in the grana, or what you call the thyroids. The combination of thyroids forms grana. So the answer is this is the grana. This is what we classified as grana. So where do we find grana? So basically it's D, which means location in D. So 2 marks in there. Then they're saying that all the questions, all questions are being combined. All right, they're saying the graph below shows the effect of temperature and oxygen on the rate of consumption of glucose and the corresponding rate of production of lactase lactate, that is ionized form of lactic acid at 4 different temperatures. So we have the 4 different temperatures. We have glucose lactate, okay, glucose consumed in absence of carbon dioxide, okay, sure. They're saying that under which of the following condition is the rate of lactate formation highest? Under which conditions? So in this case, we have to look at under which conditions 10. If you look at 10, 10, it's not but then 20 in absence of oxygen. So we have to look at that, and they're saying that is consumed in absence of oxygen, then lactate formation in absence of so it looks to be in absence of, so here it's 20 21. Yeah, so this is the one, but here it's saying that the one which has in absence, the one which has the circle and the dotted line, it is like this formation in absence of oxygen. So automatically it means that it's this one. So I'm gonna have to say lactate formation in absence of oxygen. So it is the, if you draw here a line, it is around 21. So it's 21 degrees Celsius in absence of Then the next question, they're saying that why is the rate of consumption of glucose higher in absence of oxygen than in presence at 35 degrees Celsius? Why? The rate of consumption of glucose is higher in absence of oxygen than in presence of this. Oh, okay. Stimulus directed from endop. Oxygen inhibitors to make up glucose by itself. Okay, no. The glycolysis reaction requires presence of oxygen. Less ATP is formed from the glucose in absence of what? Yes, in absence of oxygen. Whenever remember you produce two, then the other one produces 36. So there is a difference there. When there is presence of oxygen, you produce a lot of ATP. But in absence of oxygen, yes, the ATP is what is much more less. So basically D becomes the most correct answer. So who got 20 out of 20 in that section? Let's look at the terminologies and then we see what we can say. Organisms where the nuclear membrane is not enclosed in the membrane, we call it the prokaryotes. Prokaryotes. The diploid state of the life cycle of a plant. How do we call it? In the most, we call it, we say that we call it a sporophyte. Yes, the sporophyte. Then they're saying that the process during which energy is bonded to the third phosphate or the group. So how do we call it? Phosphorylation. Say that we call it phosphorylation because you add, remember it's ADP plus an inorganic phosphate. An organic phosphate, you form ATP. So ADP plus in an inorganic phosphate, we call it uh, uh, phosphor, eh? Phosphorylation. So because we're using oxygen, so it becomes oxidative phosphorylation. I think you remember that in the process of in the process of respiration. So we call it phosphorylation, process in which water molecules are split into electrons. I said that you can't go to the paper without knowing that. We call it no. It's photolysis, photolysis or photolysis. So basically that is it on that question. Let's go to question 1.3. 1.3. So um, we are saying that indicate whether each of the statement in the column, okay, sharp. They're saying that, let's just go direct to the questions because we know the instructions. Gel-like non-cellular layer in cnidarians. Gel-like non-cellular layer in cnidarians. How do you call that? We call it mesoglia. So the answer is A only. They're saying that 1.3.2, the virus that attack bacteria, we call it bacteriophage. It's B only, B only. Yes, they're saying the simple digestive system having only one open. We call it a blind gut. But even platyhelminthes also have the same, they have the blind gut. So I would say both A and B. Phylum to which coral, jellyfish, and then sea anemones belong. To which phylum do they belong? They belong to what we call nidarias. They are nidarias. So the answer is A only. So I think you got 4 times 2, which gives you 8. Then we go to the next question. The next question is about the phylogenetic tree. Okay, we talked about this still. Saying letter representing the following characteristics: tissue level of organization, meaning that we are looking at the tissue level from the cellular level to now polyphera, then the tissue level. So it means the answer is A. So the first answer here, it coordinates is A, and then blood Diboloblastic organisms, diboloblastic organisms, diboloblastic organisms. These are nidarias are diboloblastic animals, organisms. So the answer is B. So here I'm gonna say it's B. B goes A goes with A, B goes with B. And then they're saying that development of the coelom, development of the coelom. All these ones before they don't have a coelom. So it is D. All these organisms above they have a coelom. So D is development of the what? Of a coelom. So the answer is D. Jointed appendages. Jointed appendages. Jointed appendages. Yes, meaning that they have appendages. It's arthropods. Arthropods. So the answer becomes E in that regard, like insects. Yes, those are examples of the organisms which have appendages. Notochord. Notochord is a what? Notochord is um, when still in a primitive state. Yes, it is a vertebral column, is a backbone. Yes, but in a primitive state we call it a notochord. So when sometimes it develops, then it forms what you call a what, a backbone. Because we say that we came from that, remember that theory of which described that ontogeny recapitulates phylogeny, that history repeats itself, that we developed the notochords when during our developmental life stages, like when you're still in a womb, we have those notochords. So it's a sign that it's a structure which developed into a spinal cord, or what we call the vertebra. So the answer is for those organisms which have the vertebral column, which called the chordates. So the answer becomes F. So it means that the answer here is F. All right. Then we go to the next question. Give two advantages of a coelom. This one we have a lot. You can talk about allows creation of hydrostatic force to generate movement in the soft-bodied animals like the earthworms. You can also talk about that it brings about cushion. They act as a shock absorber to protect the internal organs. It helps the organisms it helps to keep the organisms in place in a structured way. So we have a lot. Yeah, we have a lot. So it depends on what you have really written. So, what is cephalization? Basically, cephalization. Yes. So we mark the first two. Yes. What is cephalization? Cephalization is the development of the head. So basically, that is when the concentration of the nervous tissue and the sense organs are at the anterior side of the what of an organism. So basically, it's the development of the head. Basically, is the development of the head, cephalization. And we talked about, and this is one of the terminology I say that you cannot go to the paper without knowing it. See that our prediction was very well. Say that it picked up to 90% of the paper. Whatever we talked about, they brought it. Then we go to the next question. Give the letter of the part that is diboloblastic. Diboloblastic, we say that it's two layers. So we say it is B. Then we say that triboblastic acylomate. Triboblastic acylomate, it is this one, triboblastic acylomate, meaning that three layers, but it is acylomate. It doesn't have a what? A coelom. And then lastly, the triboblastic coelomate, the one which has these gaps, as I told you from even a distinction material book, we say that it is the one which is mostly developed. Therefore, that is A, meaning that we have B here, we have B here, and we have C. And even we chose better questions which looked exactly or similar to this question. Yeah. All right. Those people who watched Thunder Eduk before they won the paper, thank you, killed the paper. Identify the part. Um, Identify the parts labeled 1. So we need to know what is 1, what is 2, and then what is 3, and what is 4. So they're saying that 1, we go back, we look at 1. 1 is there. So how do we call 1? 1 is the ectoderm, basically that is an outer layer. Then 2 is the gut. 2 is what you call the what? The gut. And then 3, what is 3? 3 is the mesoderm. Meso, the middle layer. Mesoderm. And then when we say 4, 4, what is 4? 4 is the endoderm, the inside layer, the inner layer, endoderm. Yes, so that is it. Identify parts labeled. Okay, we are done with that. Arrange the body plan in order of increasing complexity of body plan from the least to the most evolved by using the letters. So from the list, so I'm gonna start with the B. I go to the C, I go to the A. The way how they are arranged here is the same way how it's supposed to be written there. So it is gonna be B, C, then A. So meaning that A is the most advanced and then B is the least advanced. So it means that now, guys, now here we got 50 out of 50. Congratulations for those people who got 50 out of 50. Let's go to the second part. So the question, they're saying that we the extract of the graph answer the following questions. An example of bacteria like linked with a very serious disease called bacterium diphtheria. They're saying that so this diphtheria is a constitutive agent of diphtheria. The bacterium produces a potent toxin that can damage the tissue and then the respiratory system, leading to sore throats. We have fever, we have difficult breathing, and then severe cases on the heart. Heart, nervous, the nerve damage. Diphtheria was once a major cause of illness and death among children, but widespread vaccination had significantly reduced the impact. The diphtheria vaccine commonly given to the as part of this, meaning that diphtheria, tetanus, and then a cellular. Yes, this vaccine work by stimulating the immune system to recognize and then neutralize the toxins without causing the disease. The immunization has been instrumental in controlling the diphtheria outbreak and then protecting the public health. Right, that was obtained from there. So they're saying that what 2.1, what bacteria cause the diphtheria? So we have seen it here, the chronium diphtheria. Yes, this is the one which caused the what, but they're saying that what which bacteria cause, so it means that it must be written and underlined as a scientific name. Yes. Why? Because here it is in italics. Remember, when you're typing, it can be in italics, or in your writing, when typing, you have to underline it. How does this bacterium harm the body? How does it harm the body? So we have seen it that it produces potent toxins that can damage the tissue and the respiratory system. So we have seen it here. Yes, we say causes. We said it here. It produces these potent toxins that can damage the tissue and the respiratory system of the what of the body. They're saying that question number 2.13, saying that to point, name the two symptoms of diphtheria. So we see it here, fever, sore throat. We saw it here, fever, sore throat. What else? I don't remember. They said it here. Where is it? Causing the producing the toxin, cause leading. Yes, here. Sore throat, fever, difficulty in breathing. Yes, that is the answer for that. And then they are saying that how does DTP vaccine work? We see now that we say that here that how does it work? We said it worked by stimulating immune system to recognize and neutralize the toxin without causing the what? The disease. So we see it here. That's how it works. It works through that without causing the what? Without causing the disease. As I told you that when you're doing the prediction that the disease, when the disease comes, even if you have not studied it, you must be able to answer that. Why? Because it is just a passage. Because we don't know which district or which area, which school has focus on that specific what, that specific disease. Because one can cause talk about HIV, then another one will talk about flu as a viral disease. Mhm. Then they're saying that which type of immune system will be acquired by individual who received this, meaning that you have introduced it. So we can call it artificial acquired active immunity. That is an artificial. It is an acquired active immunity, but it is artificially done. So it is an artificial acquired active immunity. Then they are saying that explain uh, relationship between the occurrence of diphtheria and the vaccination coverage per year. So what are we supposed to talk about here? We look at this graph. They're saying that as the percentage of this increase, yes, DTP increase, as the percentage of DTP vaccination increases, the global diphtheria report cases start to what? Decrease. So as you increase the number of reported cases, you see that they're dropping. You understand? Yes. We see that as the percentage of these tetanus or vaccination coverage, we see that the coverage is increasing as these years are passing. Yes, this one shows the vaccination, while here showing the number of individuals. So it is the number of individuals goes down as you increase the number of vaccination. So it is basically supposed to look like this as one graph that this as the number of reports decrease, no, no, as the vaccination increase across these years, the number of cases which are being reported do what? Decrease. So that's what is supposed to be explained in that regard. Give one possible reason why the vaccination coverage per year decreased in 1992. Give give the give not that one, because these are number of cases, and then we see that now the diphtheria is starting to increase. Why did it decrease? Because it was going like this and then decrease. Yeah, what happened? Maybe it could be a catastrophe, disorganization of the health care. It could be there must be maybe an effect on this vaccine. Yeah, maybe there was a war. Maybe the staff were on the strike. So there is a lot of things which you can talk about, which what will disrupt the health sector, so that those vaccinations are not being what administered, so that it keeps the number growing up. So that's what they wanted you to talk about. So we also identify the plant A, B, C, A, B, C, A, B, C, A. A, we see A is where is A? A is Z. These are, is it A? Yes, these are pterophytes, or what we call the pteridophytes. Yes, pteridophyta. The pteridophyta is a what? It's a phylum. So you can call them pteridophytes or pterophytes, either way. Then we go to the B is brophytes. These are brophytes, and then C, we call them the gymnosperms, the plants with the naked what? With the naked seed. Label part 1, 1, 1, 2, 1 to 1 and 6. 1, part 1. So how do you call part 1? Part 1 are, how do you call part 1? Part 1 is rhizoids. Let me clean up here. So they are looking for this part, not this part. So if it is this is called a rhizome, and then this is called a what? Rhizoids. So this is what? A rhizome. Yeah, so is the rhizome is like the stem. It's the stem like structure. Then part 6, where is part 6? Oh, they are looking for this part. So the part 6 is called the needle leaves. These are leaves. Basically, these are leaves, but they look like needles. That's why you call them needle leaves. Then they are saying that we go to how does part 7 differ from a flower of angiosperm? Part 7. So we see that part 7 has naked seeds, while the flowering, the seeds are protected within a what? A fruit. So we call them gymno, gyne, gymno. Yes, it's because their female reproductive structure is naked. Gymnosperms. Yes, they are just they don't have a fruit. The major difference between the gymnosperms and the angiosperms, both of them are spermatophytes. Yes, they are seeds because sperm means a seed. So both of them are spermatophytes. They are seeds. So, but the major difference between the gymnosperms and the angiosperms, their seeds are embedded into a fruit, while the gymnosperms, their seeds are naked. That's why we call them naked seeds. These are plants with the naked what? Naked seeds. So that is the difference between the two. In which way? Okay, we are done with that. Provide a number of sporophyte generation of plant B. They are saying provide a number. The sporophyte. Remember, in this is the sporophyte. Now this is sporangium. Yes, and then this one is the gametophyte. Remember, in the moss, let me clear up. In the moss, the dominant generation is a gymnosperm. Is the did I say is the a gametophyte, not gymnosperm. Is a gametophyte. So the gametophyte, this one is the gametophyte, while this one is the sporophyte. So if they are looking for the letter which indicates provide the sporophyte generation in B, so the answer is supposed to be number 3. So it's not easy to be found. It only found you find it when there is a favorable condition, that part. While this one is easily be found, therefore it means that the dominant generation, the generation where this plant is always, is a gametophyte. So the answer here becomes 3. Then we go to the next question, which is okay. The next question, which is a flower. We talked about the flower, and we said that don't go to the paper without knowing it. Which flower, A or B, is windy pollinated? Windy pollinated. So the answer is A. Yes, the answer is A. Because of this, it's very feathery, so so it's too much, so therefore it's able, when it blows, it's able to fall on the what? On the stigma. And then if you look at this, the anthers are lower than the what? The stigma. Therefore, and if you look at the petals, are big enough so that the insects can what? Can land on them, and then corresponding and pollination can be and can take place. They're saying that give the visible reason why. Okay, first of all, we can talk about the flower are much more smaller than the insect pollinated flowers. I've talked about it, that this one is big enough. And then we can also talk about the large anthers. The anthers are large enough so that they're able to when it's blown, too much can fall on the stigma. Then we can talk about the anthers have long filament that can cause the anther to hang outside. You see, the stigma are long and feathery. Yes, the stigma are what? Are long and what? And feathery. So that you're able to attract. They're able to what? Do what? To attract that. They're able to attract it. So we see that our anthers are very long, and then we see that the stigma is able to capture that when it happens that there is any blow of what of wind. Then we are going to the next question. Give the visible. Okay, identify part three. We see that it is a filament, and then part six, these are called the sepals. Sometimes they call them um sepals. Yes, these are the green parts which actually encloses the flower when it is there is a difference between sepals and the brackets. Yes, brackets. So they are not the same. Sepals are green. They try to protect, bring about the protection. They are connected to the receptacle. Yes. Just in most cases, most of the flowers, it is the outside part of the flower where the flower join together. They're saying that give the number or the parts of that produce pollen grains. Pollen grains. So here we can say that is anther, name or then they're saying label. Yes, we can say it's anthers which produce pollen grains. So here we can look at anthers, it's number 2. Then you're saying that receives pollen grain during pollination, receives. We can talk about the stigma, it is number 10. And then you can also talk about the stigma, which is feathery here, it's number 9. So this is the stigma, and then these are the anthers. And then we are talking about which develops in the fruit. Develops in the fruit. What develops in the fruit? Develops in the fruit. They don't understand that. Which develops into the fruit. Oh, correct. Develops into a fruit. This is the ovary. Yes. So the answer here is ovary. Here we can't see anything here. And then you're saying that basically it's number 4. They're saying that develops into a seed. These are ovules. So we have to look for the ovules, which is the number 5. Let me clear it up so that I'm able to see clearly. I don't want to give you a wrong answer. So the answer, yes, it is ovules, which is the number 5. Then we go to the next question. Explain the ovulation advantage of angiosperm have over pteridophytes. Angiosperm, that is the gymnosperms and the angiosperms. Not the spermatophytes. The angiosperms, the fruit-bearing plants, have over the pteridophytes regarding to their reproductive dependence. So basically here you can talk about that the angiosperm don't require water for reproduction, while pteridophytes require a film of water for reproduction. So you can talk about uh angiosperms, they have what called a pollen tube, so which allows it to the pollen grains to go through into the ovary. While the pteridophytes, no, not the case. The sperms have to swim through the water so that it's able to reach the what? The ovary. You understand? To not the ovary, okay, to reach the female reproductive what? Gamete. So this it helps the angiosperm to survive in harsh conditions. Yes, which is not the case with the pteridophytes. So if you require water for you to undergo reproduction, if the water is not present, then what happens? Then it means that what's going to happen is you don't have, you don't have, you can't carry out reproduction. But the angiosperms, whether the water is there or it's not there, the reproduction will take place. Yeah, so that is a major advantage over the other one. So we have to read another question 2.4. A group of researchers investigated antifungal treatment in most inhibiting the growth of fungi. Label the petri dish with the name of the antifungal being tested, and one of the control case spread the fungal spray evenly over the surface of each dish. soaked papers discs each fungal solution, and then place the disc in the center. Okay, place the disc soaked, okay, sterile, and then water in one petri dish, and then seal the petri dish in incubate at that temperature. After 5 days, observe the growth, measure the zone. These are the things which you did as an experiment. Then they are saying that, what are they saying? That name the three planning stages. Here you have to plan. You have to plan, decide on, decide on. Always start with your answer, decide on, decide on the type of fungal that you're gonna be using, which decide on which growth medium they are going to use, decide the apparatus which are going to be used, decide on how the growth is going to be measured, decide on how they are going to record the results, decide the temperature on which the growth is going to be taking place, decide. So the word must start with the word decide. When it comes to the planning stage, the word decide on is supposed to be there. Please, 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 so that you're able to get the marks. They're saying that identify the independent variable. So if you look at the independent variable, it shows that here the antifungal agent, yes, will determine the growth, basically the growth. Let's go back to the question which they said. A group of researchers investigated on how the antifungal treatment is most effective at inhibiting the growth of the fungi. So meaning that this one is the one which determines the growth of the fungi. If the type of antifungicide is the one which is strong or in inhibiting, then it means that the growth will also be very small. So the answer there, we can say that the growth of fungi, and then here it is the antifungal, it's the antifungal agent, which agent they have used. Say that describe one way in which the ability of this investigation could be improved. How should they do to improve this investigation? Basically, you have to conduct this experiment multiple times. For each antifungal agent, meaning that each antifungal agent is supposed to do multiple times. Yes, and then they say that they used something petri dish. They use over the spray. They put the number. Where did I see it? Uh, they put the number of, they're saying the surface of the, I don't remember where, so it had, they said it here, label them for, use more than four, use more than four petri dishes. It can also be working in that area and then determine the average. So we use more than four and then determine the average. Name two factors that must be controlled to ensure that the validity of these results is there. So here we will talk about the amount of fungi, amount of agar, the amount of antifungal, the type of fungi, all of them, yeah, there must be what? Must be the same, must be kept constant so that you're able to control. Yes. State the conclusion of this investigation. So we can talk about the conclusion of this investigation. You can, so yeah, we have to look at which one is most effective, because here this shows the inhibition, the percentage, like the inhibition. So it means that clotrimazole is the most effective antifungal agent at inhibiting the growth of the fungi. Yes, this is the most effective fungal agent in inhibiting the growth of what? Growth of the fungi. So basically, that's it. And then you get 50 out of 50. Then the last question, we did. Yeah. All right, let's just discuss. There's nothing I can say because the way we predicted it 2 weeks before is the same way how the paper was. And then thank you so much for those people who watched our videos before they went for the paper. I think now you can witness that Sunday Duke is the best. All right. They're saying that a plotted plant with variegated leaves was discharged, and a strip of black paper was used to secure over both sides of the leaf. The plant was exposed to sunlight for 4 hours. The leaf was removed and then tested grains of starch. Okay, saying why is this necessary to discharge the plant before the investigation? Why should I? To ensure that starch formed is because of the factors which is being tested for. So we discharge the plant so that there is no any other starch which is there. So that any starch which is being formed is as a result of the factor which we are investigating about. Provide a suitable aim for this investigation. I can say that because this thing is about light, I can say that to determine if light and chlorophyll is necessary for photosynthesis. Yeah, because of these parts. Yes. So to determine whether chlorophyll and light are necessary for photosynthesis. Then we are going to the part or leaf which part or parts of the leaf act as a control experiment. So here, the control experiment is the part of the leaf, the part of the leaf covered with the black paper. The black what? The black paper. So it's the one to show that light is basically necessary for photosynthesis. So question 3.2, they say in the diagram below show transverse section of the leaf that study the answer and try to answer the question. We see that these are the epidermal cells. Then we have this peristeid mesophyll. We have the spongy mesophyll. Then we have here the vesicular bundle. Vesicular bundle. Then here we have the what we call a Segadi cell, and the whole there we call the stomata, the stoma. The stoma, not stroma, because stroma is found in the chloroplast. Identify part B. We say that part B is the vesicular tissue, or what we call the phloem. Then you're saying that part C, we say that part C is the stroma. This, no, I mean not stoma. Yes, which we call the, don't call them the guard cells, because the guard cells are the cells which guard to allow it to open and close. So basically, these are the cells. They're not asking you about the cells, they're asking you about the whole. Yes, so it is what you call the stoma. State the importance of part C. Part C, this one is very important in allowing gaseous exchange, meaning that carbon dioxide in and oxygen to exit or out. Tabulate the difference between part D and E. So here, when you say tabulate between part D, so I told you that draw the table, draw the table. If you don't know, at least you draw the table. Then you say, don't just leave it like that. You get zero. Put that part D, part D, and then you say part E. If you don't know anything there, then after that, that will give you a mark. Then now you can list anything there. So if you look at this, we see that this black thing, remember these are chloroplast. So this one has too much chloroplast, fewer chloroplast. Then you can talk about the cells here are fully packed in the peristeidal mesophyll, while in the spongy, they are loosely packed. You can talk about here the cells, they have irregular shape, while here they have regular shape. We can talk about that. Here there are too many air spaces. Here there are small air spaces. You can look at some of the difference between the two. Then we got the next question. They're saying that study diagram, diagram of aerobic respiration and answer the question. So let's go to the aerobic respiration and then we answer those questions. So we see that you have oxygen coming in, and then you have you form two glucose. No, not oxygen. You form two what? Six carbon molecule, meaning that that's glucose breaks down into two, which is pyruvic acid or pyruvate. Then the pyruvate acid enters the it enters the what? It enters the mitochondrion, and then the Krebs cycle happens. And then after that, it undergoes a chain reaction, which we call the oxidative phosphorylation. And then water molecules and ATPs are being what? Are being formed. So we see that. Let's find out what is it, and then or what they want. Which substance is indicated by A, B, A, and B? We see that. Let me check and see. Okay, 3.3.1. They're saying which substance indicated by A, we say that that is glucose, and then which substance indicated by B, it is pyruvic acid, or what you call a pyruvate. That's what I said. Then we said that identify the stages A, B, C. Stage A, where is A? Stage A, we say that this is glycolysis, and then stage B is Krebs cycle, and then stage C is oxidative phosphorylation. Then they are saying that identify molecule X. Molecule X, that is we go back and molecule X, which forms between phosphorylation. So how do you call that? We call it molecule X. Molecule X, it is ATPs. This is ATP. ATP. Yes, it's ATP. Where is the next question? Oh, identify the gases. That is 1 and 2. Identify gases 1. 1. Where is 1? Where is number 1? What is, I don't see 1 and 2. My God. Oh, the gas is here. It's being produced here. 1. Is number 1. And then gas 2. Where is 2? I don't see gas 2. It's gas 2. Let me first clear up, and then we see. Remember, it's a respiration. Since there's a respiration, the gas which is being produced, yes, it is carbon dioxide. Carbon dioxide. So it's carbon dioxide. You write carbon dioxide in full. And then where is 2? Okay, let me scroll down to check and find. Oh, it is here. I couldn't see it. So yes, it is. We use it. Remember, it is oxidative phosphorylation. Phosphorylation means the addition of ADP plus an inorganic phosphate, but it happens in the presence of oxygen. Therefore, we call it oxidative. So that gas too, it is oxygen. So it is, this one is carbon dioxide, this is oxygen. So we have answered that one. Why does 3.3.5, we are saying that why do comrade athletes often get muscle pain when they and the cramps? Basically, this is because of the insufficient oxygen, which makes the production of what? Of lactic acid. It is because of the lactic acid has built up due to, as a result of anaerobic respiration. Yes, as a result of absence of enough oxygen. Basically, the aerobic respiration is incomplete. Remember, anaerobic respiration is as a result of an incomplete processing of this glucose to break it down into carbon dioxide, yes, and ATP. You understand? But now, because it is there is not enough oxygen. This, to completely do that, you require enough oxygen to that to happen. It's like when you burn carbon in air, you produce carbon dioxide. But if you burn it without oxygen, like there is insufficient oxygen, so you produce carbon monoxide. So carbon monoxide is lactic acid, and then carbon dioxide is what? Is when there is complete, because when it is complete, it means that the reaction is gonna completely be broken down, or it's gonna be processed completely. So if there is no oxygen, it means that it's going to only stop here, and then now the pyruvic acid and the pyruvate is going to be turned into lactic acid. Since it is an acid, it brings about those cramps and also paining of the what? The muscles. Then we got the next question, which is question 3.6. Draw the elaborate structure of the cellular respiratory takes place, including the names and organelle. Or you see, this is a mitochondrion. But you don't go the paper without knowing a mitochondrion. Just draw an outer layer which is smooth, and then the inner layer is infolded. Some people, when they are drawing, I don't know what they are drawing. They are drawing structures which I don't understand. Yeah, and then you put DNA, you put ribosomes, you put the crystal, you put the matrix, you label the caption. Some people, they just draw like this. What is this? Huh? Guys, something properly. Eh? I don't know which books are using, you guys. You must draw proper things, which don't just draw as if it's a web. This is not a lake. Yes. Anyway, um, then you have another extract we need to read, and then we get some questions from there. So we're gonna just go to the questions and then find out what are they talking about in that extract. What type of food took long? I can't answer now. Time it takes to digest. Okay. Now here we are able to see that, ne? Hmm. What type of food? Mhm, mhm. What type of food that took took long longest in the man's stomach? Man's stomach, we see here it is. Oh, it is boiled eggs. We see here it's boiled eggs. So the answer is there. Then they are saying question two, digested the fastest in the stomach. Digested the fastest. We see that it has lower number of minutes, which is raw eggs. And then they are saying that what are the main components of gastric juice involved in the digestion? Basically, we have to read here. Eh? But gastric juice, you'll find out that you have HCl, hydrochloric acid, you have water, you have mucus. Yes. You have pepsin or pepsinogen. Oh, we can, because sometimes we don't talk about a pepsinogen, we can talk about proteases, the enzymes. Remember that in the stomach, we have digestion of proteins, and the enzymes which digest proteins, we call them proteases. Yes, we call them what? Proteases. I don't want to go to the distinction material book now to show all those things, but everything is there. Then they're saying that at what temperature should the test tube be kept? What temperature is the test tube should be kept, and give a reason for your answer. Basically, it is 37. Why? Because that's the optimum temperature for the body. And then you'll find out that it means that it will be mimicking that one of the human. So that's the optimum temperature for the enzymes to work best. Did they talk about it in the passage? I don't know. But that is supposed to be as a result of that. Then they're saying that question 3.34, how would digestive process be affected if the stomach were removed after an instance? Explain. And after an accident, explain your answer. If you don't have the stomach, then what happens? It means that you have to eat less, and then you have to make sure that you chew your food very well without, because you don't have the stomach. So whatever you swallow is supposed to be absorbed. So digestive will still take place, digestion will still take place, but because we only talk about the stomach. So if it is the stomach, it means that now other parts of you won't have proteins. And make sure that what you're eating is clean, because now they won't be killed by the hydrochloric acid in the stomach. So the digestion still take place, okay, and it will still take place. That is in the small intestines, eh? Yes, but it will be much more less effective because you don't have the stomach. And then there will be increase in infection or the digestive tract because you don't have hydrochloric acid to kill the bacteria. You can also talk about um, when your digestion, the stomach is not there. So what does it? Okay, you can basically say that digestion will take place, correct? Yes. However, it will be less effective. You see? Yes. And then you can also talk about there will be an increase in the risk of what? Of infections. Why? Because the hydrochloric acid is not there. If the hydrochloric acid HCl is not there, it means that the bacteria won't be what? Killed. You understand? Therefore, you are going to be susceptible to as these infections which come with what? With the food. So that's why I say that make sure that you eat food which is very, very, very clean. However much clean it is, bacteria are everywhere. So it can be infected with the bacteria before it is swallowed. Ish. We just pray that we don't get this condition. Say that 3 point, okay, what lifestyle changes? What does the role of intestine? Okay. 3.4.5. What role does the small intestine play in the digestion after stomach is removed? That's final digestion and absorption. Basically, the small intestine is final digestion and also absorption. There's nothing much you're gonna talk about there. They're saying that now draw the background. Hey, the question is done, eh? Oh, what lifestyle changes are necessary for someone living without stomach? So basically, you have to eat small. You have to chew very well. That's what I talked about. Eat more proteins so that you're able to develop your body. Then do not drink liquids with the meals so that why to prevent this mixing. Yes. Stay upright after eating because now the stomach is not there and then now the esophagus is not there, so you have to stay upright after eating so that it can go down. Remember, these things are controlled without your will. Yes, so we need to talk about that. Draw the bar graph to represent the results. Which results? So if you are going to draw, this is what I explained, that now if you are going to draw a bar graph, it must have the caption, so we know that. Then now this is going to be the x-axis, and then these are going to be the y-axis. But I said when you're plotting such, you plot this and this. It means that it becomes one graph, and then now this and this. Then you can join it there, or you can leave a space there. Then this and this, another, and then now this and this. And then now you can do whatever you want to do, and then you can do whatever you want to do, and then you put a key, and then you tell us what is this, and then what is this. So you complete all of them, and then you put the scale, the scale here, you put the units, and you also put the what is it? Is the time taken in minutes. It's supposed to be there. And then here it's the type of food. And then here the caption must have the two variables. Make sure that the bars are the same size. The bars are the same size. That's it. And then you get your 150 out of 150. Actually, based on our prediction, we see that TO9 between 90 to 98% of our prediction, it was correct, which was released um, uh, plus or minus 2 weeks before the paper was written. Yeah, we always do the predictions, and the predictions are always on the point. And then some people, they see that the prediction is 1 hour, then they don't want to watch it. You're doing a disservice to yourself. Make sure that you watch all of it, because we are preparing you for the exam so that you're able to pass that exam without any questions. Thank you so much, guys, for joining us. It has been M. Saidi. See you again in the next